All this is Dr. Mobeen Sayed from Drbeen.com. Welcome to one more show. Today is a very important and exciting preprint study, small pilot study. And most of that study is in vitro. There is part of it that is in vivo as well. Why this is exciting is because I think that for the first time, we have come across something that may be useful, which may be over the counter and potentially help us to reduce the cytokine storm that causes people to end up in hospitals or in ICUs. So because of that, I'm quite excited about this one. The credit goes to Dr. Dr. Paul Marek of Math Plus protocol. I had been communicating with him for requesting him to join us once again and talk about his Math Plus update. And he sent me these studies. So again, credit goes to him. I want to read to you before we start his response to me. So I had asked that would he be available to talk with us about Math Plus, and he said, "Dear Great Bean, sure, we should chat about this. I strongly believe that most clinicians do not have a good understanding of the pathophysiology of this disease. If you don't understand the disease, how can you treat it? I have attached our updated protocol and Math Plus publication. I will follow with some more interesting papers and COVID-19 PowerPoint with highlight some critical points. Your faithful soybean, Dr. Paul Marek." So he's such a great guy. I really like him. So let's start. The takeaway from today's discussion are two things. Number one, the takeaway should be that it becomes clear to us that what is the role of interleukin 17 in the cytokine storm. That is one. Secondly, the role of thiamine, which is vitamin B1, the role of thiamine in calming down the interleukin 17's production, which in turn will calm down the cytokine storm. This is the outcome of the discussion today. I have done the thiamine discussion in detail once before, so I would link that after this discussion. I would link that. The link is also in the description too. So if you wanted to do a deeper dive in thiamine mechanism, then please watch that video too. So let's start. All cool beans. Let's start. So two things to note today. How does interleukin 17 play for cytokine storm, and what is the role of thiamine? And how has the study proved it? So let's start. Look at this. Their basic outcome was that the thiamine administration can attenuate the production of interleukin 17. Interleukin 17 in turn is responsible for cytokine storm. So let's see how. So we have been doing this from some time here. So look at this part of the diagram. This is a dendritic cell or a macrophage. It is innate arm of the immune system that takes up a pathogen, and then what it does is, we know it connects that with a naive T cell, correct? And then in the presence of interleukin 4, the naive T cell becomes T helper 2 cell, which then releases interleukin 33, 10, 4, and 5. The interleukin 4 and 5 specifically act on B cells, which then become proliferated as a result of interleukin 4 and 5. They also do class switching to go from IgM to IgG and so on. And they start producing antibodies. We know this part. The second mechanism, the second pathway of a naive T cell is that in the presence of interleukin 12, which normally is secreted by the dendritic cell or macrophage, the cell that ate up the pathogen also secretes interleukin 12. And in the presence of interleukin 12, we go to T helper 1 pathway, which will then cause interleukin 2 production, the T helper 1 cell. That cell would then activate the cytotoxic T cells, and then cytotoxic T cell would start killing the cells that are infected. We know this, I think, quite clearly. Now, we have done this discussion a couple of times. So I'm going to just review that because this is the more important part in today's discussion. This is about the T regulatory cells or T helper 17 cells. In the presence of interleukin 23, the naive T cell converts into T helper 17 cell. T helper 17 cell in turn releases multiple cytokines. Cytokines are the small hormonal molecules that are released by the cells to influence the other cells. Those chemical molecules released by these cells that attract the other cells to the area of the infection are called chemokine. Chemokines are cytokines as well. So here, if you look at this, the T helper 17 releases GM-CSF, which is granulocyte monocyte colony stimulating factor. What that means is this molecule would go and act on bone marrow to increase the production of granulocytes and the monocytes. That means eventually we'll end up with neutrophils and macrophages and basophils and eosinophils and so on. Interleukin 17, this is the hero molecule for today's discussion. Interleukin 17 is also released by T helper 17. Interleukin 21, interleukin 22. Interleukin 22 is another molecule that we would discuss today, maybe not in that much of a focus, but we'll discuss that. The difference between these two, I'll spill the beans right now. Interleukin 17 is pro-inflammatory. It triggers, it promotes inflammation. Interleukin 22 is anti-inflammatory. It tries to suppress the inflammation. And then tumor necrosis factor, interleukin 6 are also released by T helper 17. Then T helper 17, when it releases interleukin 17, and my apologies, for my throat, let me show you a couple of things before we continue. This is the situation out here in my home. This is how bad it is. These are some sort of clouds that are formed. So this is the smoke clouds. And look at the ash. So these are my cars, and look at the ash on them. So this is the result of the fires and the smoke here. So it kind of irritates my throat. And while we are at it, let me also show you this. A cool bean, or actually two cool beans, Marita and Frank, they sent so Luffy and Kyrie are our little pets. So they sent their Snoopy and Charlie. So here these two little rascals are, look at them. they're playing with the stick here. So wanted to show that as well. So back here. So interleukin 17 causes the epithelial cells or endothelial cells, so that means nearby tissue cells, to release GCSF, which is granulocyte colony stimulating factor. GCSF is a molecule that would go and work on the bone marrow and make the bone marrow make more neutrophils. Epithelial or endothelial cell under the influence of IL17 will also release interleukin 8. Interleukin 8 in turn is a chemotactic factor for neutrophils. It attracts the neutrophils to the site of infection and inflammation. Secondly, it also activates the phagocytic cells, interleukin 8, and asks them to do more phagocytosis, break down more pathogen and break down more cells. All of these behaviors or methods or mechanisms are going to increase the inflammation. How? By making more inflammatory cells, by calling them into the area of inflammation, by asking them to be more active. Then if you look here, this interleukin 17 here is also going to go back all the way and act on the innate arm cells, that is macrophages and dendritic cells. And what it would do is it would ask them to also release GCSF and interleukin 8. In addition to that, when the innate arm cells will become activated, then what they would do is they would release their own cytokines. Out of those, what are important are IL-1, IL-6, interleukin 8, tumor necrosis factor, GM-CSF, and IL-12. We have done this talk in the past as well. Interleukin 6 here is important to note as well. And that is, number one, we all know that interleukin 6 helps with the fever and temperature increase during the infections. Interleukin 6 also goes and works on the liver and causes the liver to produce more proteins for inflammation. These are called acute phase proteins. Interleukin 6 also causes increased production and activity of neutrophils. So if you can see here, what is really happening is that from the innate arm to the naive cell to T helper 17 to the production of interleukin 17, this one axis gives rise to production of lots of cells, inflammatory cells, lot of inflammatory molecules, and lots of inflammatory activity. The point they're making in this study is that if we can block interleukin 17, then the production of a lot of neutrophils will not happen. Production of a lot of GCSF and IL-8 and then interleukin 1 and interleukin 6 will not happen. So the drugs like anakinra or tocilizumab may not be needed if we can take thiamine, because thiamine is going to try to suppress the upstream interleukin 17, which then in turn gives rise to interleukin 6 and interleukin 8 and other things. That is their basic point. The question for them was how much dose to give. So I hope that this mechanism is clear here. This axis is clear. And if this is clear, I'm going to move on to the very quick review of thiamine. And we have done a discussion about thiamine. I'll link it here as well. And the link is present in the description of this video as well. So high dose thiamine, 100 milligram, what they did was in the study, they had given 100 milligram BD. That means 100 milligram twice daily. So that means 200 milligram per day thiamine. It is safe, which they proved in this discussion. It is cheap and it is over the counter. Imagine trying to go and find anakinra or tocilizumab or remdesivir, and imagine compared to them just going to go get thiamine. Thiamine, they proved, reduces the interleukin 17 production. So if it would reduce the interleukin 17 production here, then all of this mechanism will be suppressed. All of this mechanism will be suppressed as well. So a lot of pro-inflammatory actions will go away. That's what they are proving. Now, thiamine, we know, is a coenzyme with dehydrogenases. This is a quick review. I've done a detailed discussion. Thiamine or dehydrogenases with thiamine, they take part in energy production. So if we cannot do proper energy production, then what would happen is, in the absence of thiamine, instead of going into the Krebs cycle of energy production, we'll start making lactic acid, which will cause acidosis. Antioxidants are produced in our body all the time. Thiamine helps as an antioxidant. Thiamine is very, very important for DNA and RNA production. And in the absence of thiamine, I think we all know that we start getting megaloblastic anemia and we start getting the neurological symptoms. Branched chain amino acid metabolism is also helped by dehydrogenases, especially with the thiamine. Triglyceride breakdown, so some of the lipid types, their breakdown also needs thiamine. So these are just generally the uses. So now let's look at the study. What did they do to prove that high dose thiamine is useful? I love it. Thiamine is over the counter. So here is what they did. They took 16, and again, this is a preprint. This is a pilot study. It is a small study. It is a combination of in vivo and in vitro. And the suggestion by the authors was that we should now do larger studies. So they took 16 folks who had alcohol use disorder, who were known to have been drinking a lot. And they gave them 100 milligram thiamine BD for 3 weeks. So 200 milligram thiamine daily. They also had 8 control subjects who were not alcohol use disorder patients, and they were healthy. After giving thiamine, they observed their interleukin 1, 6, and 10 levels, the authors, and the authors also measured their interleukin 17 and 22 levels. What they found was that in this alcohol use disorder persons, and the question is, why did they use alcohol use disorder patients? Because these patients, these persons, have a lot of inflammation in their body, and they produce a lot of interleukin 17 in their body. So they wanted to, so that is a very similar mechanism as what happens in COVID and septic shock and other diseases. So you can say that alcoholics are more prone to inflammations, and so they used them as a study group. So they found that interleukin 17 and 22 production was reduced when thiamine was given. The age group was 21 to 65 years. So now let's see so this was the in vivo part, that is in persons. This is what they did. Then they took these studies data from these people, and then they continued with the study in vitro, that means in the dish. Why did they do it? They wanted to directly work on the cells and put thiamine and see the safety of the thiamine. They wanted to see the dose of the thiamine. So they didn't want to do all of that on human beings. So they gave thiamine to human beings. They looked at the response in them. Then they verified the response with different dosages and with different mechanisms or dynamics of their pharmacokinetics, and they came back with the results. So this is the in vitro part of the study now. What they did was they took some mouse macrophages. So these guys here, the ones in blue, these are macrophages from a mouse or mice. To them, they administered 0.01 microgram per milliliter of thiamine, which is equal to 100 milligram per day thiamine dose, not 200. To humans, they were giving 200. To these cells, they gave equivalent of 100 milligram per day. Once they treated them with thiamine for 2 hours, then they further treated these cells with ethanol. They made them drunk by pouring ethanol on them. And they kept these cells now in ethanol for 22 hours. So 2 hours in thiamine, high dose thiamine, then 22 hours in alcohol. Then what they did was they gave these cells, they took them out of that dish, they gave them a shower, they cleaned them, they washed them. So the thiamine is washed away and the alcohol is washed away, and the cell is now once again clean. Then they went into the cell and they extracted their RNAs. So what they did was they wanted to see that what kind of gene expression and what kind of RNA is produced inside the cell as a result of thiamine, excuse me, and as a result of ethanol. So the result was they collected the RNA samples, which showed interleukin 17 and 18 RNA expression. Then what they did was they created a reference. I wish that somebody used to make these diagrams for studies for me when I used to study. This makes it so much easier to understand. So then what they did was they took interleukin 17 profiles of various kinds of patients. For example, they looked at the interleukin 17 levels in healthy individuals and created a range that in healthy individual, interleukin 17 is from here to here. Then they looked at the patients with metabolic disorders, for example, vitamin B deficiency or with alcohol abuse syndromes. These are called metabolic disorders. And they looked at the interleukin 17 range in these patients. Then they took the literature for septic shock patients and they searched for what kind of interleukin 17 range is in them. Then they took viral infection, included COVID-19, and looked at the interleukin 17 range in them, and they created a chart. So now they had a chart for what kind of interleukin 17 ranges are observed in various patients and compared to healthy. So of course, then their target would be to give thiamine and see that can they convert a range which is high to a range which is towards healthy. And that is what they came back and they said, yes, we were able to do that. So here were their results. What they saw was in the human part. So remember, this study had, this is a preprint, has two parts, a part run in humans and a part run on cells. In the human part, they saw that the interleukin 1 and 6 was high in alcohol use disorder syndrome patients. They also saw that interleukin 17 was elevated in the alcohol use disorder patients and not in the healthy patients or healthy persons. Then they saw that when they gave thiamine to these 16 individuals, 200 milligram per day thiamine, the interleukin 17 quantity reduced by 4 times. So there was a suppression of interleukin 17 production, which is excellent. That's what they wanted to prove. They also saw interleukin 22, that did not change too much. And interleukin 22 is anti-inflammatory. We want more interleukin 22 and we want less interleukin 17. Both of these are produced by T helper 17 cells. But in case of viral infections or alcohol abuse, these cells, the T helper 17 cells, become dysregulated. They start making more interleukin 17 and less interleukin 22. So the pro-inflammatory behavior increases and anti-inflammatory part is suppressed. Here what they saw was that in the presence of thiamine, interleukin 22 levels did not go down, which normally should have gone down in alcohol abuse. So that means somehow thiamine was able to keep it at the normal level. It didn't increase it, but keep it at the normal level. It rescued it. On the other hand, interleukin 17 was high and thiamine was able to bring it down by 4 times, which is excellent. Love it. So interleukin 22 and 17 was observed. Then inside the cells, that those macrophage cells from the mouse on which they were putting alcohol and trying to make them drunk, the cells part, what they saw was that thiamine reduced interleukin 17 and it rescued their interleukin 22 levels. That means that when they put the alcohol on these cells, the cells were going to produce more interleukin 17 and less interleukin 22. However, in the presence of thiamine, that didn't happen. So so these drunk cells actually stayed okay. Then finally, what they found was on these cells, they did various kind of dose related. So on these guys, they did various dose related activities on them and did pharmacokinetics to figure out what is the range that is safe for the cells. So in their experiment, they found out the range in which the cells did not become damaged at all. And then they did statistical analysis and they found out 79 milligram per day to 474 milligram per day of thiamine showed that it could maintain interleukin 17 levels in normal ranges without being toxic to the cells. So that is what they found out. There is a question from Luke Caspi. Would thiamine replace budesonide for fast-track treatments? Here is how I would say, look, steroids are the king of everything, right? So they suppress everything to a great extent. Budesonide is in the early part, it is given by inhalation only to the lungs. So thiamine would help as well. Budesonide is going to calm down the whole inflammatory and immune response, which may actually can become negative as well if the virus gets a free hand, immune system is not working and it just goes on causing damage. But I'm so sorry, the smoke is so bothersome. But for the budesonide, we have not seen that from Dr. Bartlett and his colleagues. We have not seen it. It actually works. I have not seen a negative effect of steroids with COVID in the beginning. So to answer your question, I think thiamine should be taken. All of us should take thiamine. It is actually good for our mental health or the neurological health. It is good for our cell production. It is good for RNA and DNA production. It keeps us healthy, and then it can keep us away from developing inflammatory responses. So especially, for example, in the US, where the only response our health authorities have is that if you have symptoms, stay home. So that means they're taking a bet on us to say that 81% are just going to become okay. The remaining 19%, we're just going to let them become worse before we treat them. So maybe having things like thiamine would help. Maybe steroids are not going to be available that much. So Luke, that is my answer. So this is the discussion. And one final point that I want to make from their discussion is very interesting. So this is the preprint here. I spent a good day today to look at it. One more thing here is here. I believe it is here. So basically what I was going to talk about was here, the neurological symptoms. What they have said is this: that the neurological symptoms, here is that part. What they did was they looked at the literature for the COVID-19 patients and looked at their neurological and cardiovascular effects. And they tried to map them to the thiamine deficiency effects on heart and neurological system. So remember, thiamine can cause wet beriberi or dry beriberi, plus the thiamine deficiency. Plus thiamine deficiency can cause Kwashiorkor, Warnicki-Kwashiorkor syndrome as well. So they found out that for the COVID-19 patient, the type of neurological symptoms were very similar to the kind of symptoms due to thiamine deficiency. So having thiamine in general is also going to help with those neurological symptoms and cardiovascular symptoms as well. So they are saying that not only it can help with reducing the cytokine storm, it would also help reduce the neurological and cardiovascular effects as well. So that is a discussion for today. Tomorrow, I want to talk about the cracking or fracturing teeth in COVID-19 patients or during the COVID-19 time, even in healthy people, plus an update on vaccines. So these are the two topics I want to talk about tomorrow. So I'll now look at a couple of questions and then we'll stop. You take care about smoke, and thank you, Margaret. Last weather outlook, the podcasting watched a laser-like beam, maybe thought around fires. Of course, they think it's reason. Maybe it's a yeah. So there was a gender reveal party that caused the fires, and so that is continuing. But thank you very much. Thank you, Chantal. Budesonide has helped my long haulers issues. Steroids have helped the long haulers a lot. Absolutely. We would benefit and have more effective, I wonder. We can check that out. Thank you, Adil. And then, yeah, so thiamine has already been given to many other inflammatory disease patients as well, for example, psoriasis. So did they find any part of the cell that was fever reduced by alcohol? Just curious. So the because interleukin 6 production reduced, that would reduce the fever as well. So interleukin 6 production got reduced, for which we wanted to have this tocilizumab. So you don't need tocilizumab if thiamine actually works out. Similarly, interleukin 1 production was reduced, which is anakinra, which blocks that. So that would also be not needed if thiamine works out. And so, yes, interleukin 6 in turn causes the fever, and interleukin 1. So they would go down. Luke says, would thiamine replace budesonide? So I just responded to that. Time Warp says, here I'm trying to scroll the paper. My apologies. I have the link to the paper. So perfect. So this is what we have for today. My request to you, as usual, please like, subscribe, and share. If you wanted to donate for my work, there is a link in the description as well for donations too. So thank you very much, and I'll see you tomorrow.